Dear students, let us discuss another topic, metamerism in annelids. So, as we have already stated that this is the first phylum which is having true segmentation on the body, and the segments of the annelids are called metameric segments or metamerically segmented body. So, let us discuss what is it. So, when the segmentation in bilateral animals you must understand animal which can be divided into two equal lateral halves involves a longitudinal division. You can see longitudinal division of body into linear series of similar sectional parts. It is termed as metamerically segmented. So it means if a longitudinal body is in turn divided into many parts, this is called as metamerically segmentation, metameric segmentation, or metamerism. Each part is called the somite or metamere or segment. That means part after part. So you can see this is a part and then there is a next part. Each metamere, each segment that I have shown you typically repeat some or all of the various organ units. So body has different organs. All organs may not be repeated. Some organs are repeated. And particularly the repeated the repeated organs are from the if the from the mesodermal origin. The term metamerism is applied only when the organs of mesodermal origin are so arranged. That means they are arranged segmentally, repeated in each segment. Then it is so said. The primary segmental divisions are the body wall musculature. So look at the division. First, it's a body muscles are the muscles of the body wall, which shows the division because of the segmentation. And then you may see that the serum, the cavity present inside it, may also get divided. Once the body wall in the cavity is divided, then uh, corresponding metamerism can also be seen on various system like nervous system may also be repeated as you have seen just prior to it during the silent features that the ganglia may be present in each segment. Cerebral ganglia present in the head, then the ventral nerve is coming out. The ventral nerve then have the segmental ganglia in each segment. So that is the repetition. That you can say the repetition of the nervous system. Blood vessels may also get repeated, and the excretory organ like the nephridia you have seen, they may also get repeated in the each segment. Longitudinal structure such as gut, because we have seen that the gut in the annelids is a long, elongated straight running from both from the anterior end to the posterior end, opening through the anus. So this gut and the principal blood vessel, main blood vessels coming out from the heart, and the nerve that is coming from the brain, the ventral nerves, they are not divided. They extend entire length of the body. But what was repeated in the nervous system? It was the ganglia, while the other organs such as gonads are repeated in each or few segments only. In addition to this, gonads may also be repeated. They may be repeated to few segments, may not be present in all the segments, but they are repeated. So it means in metamerically segmented animals, some organs are repeated in every segment, some may not be repeated in every segment. Metamerism is always limited to the trunk region of the body. As you can see, this is a typical annelid shown over here. The anterior most this segment which bears the mouth called the prostomium, and the posterior most this segment called the pygidium. They are not the segments, so the body present between them is the trunk region, and the metameres are always present in the trunk regions. Segmentation may be metamerical segmentation may be, or you can say the segmentation may be external as well as internal. As you have seen in annelids, arthropods also you may see, and the vertebrates, all they have the metamerism, segmentation of the body. Vertebrates, we are the example, best example. If you just touch your vertebral column, you will see that the bones are repeated from a neck downward up to the end. So this is the best example of the segmentation. In annelids, metamerism is both external as well as internal, so-called metamerically segmented animals. Its body consists of greater number of internal and external segments and all the body organs such as musculature, CT for locomotion, blood vessels, nerve ganglia, excretory organ which were called the nephridia, and the gonads. They are repeated segmentally. Because why they are repeated segmentally? Because internally the body is also divided. So once the body is divided internally, then some organs are repeated in each segment. Even the coelom is divided into segmental compartments. We have already seen that coelom is also divided. Now, only the digestive system remain unaffected, which is a straight long tube whose organs are not repeated from one segment to other. Now, this was the the annelids were showing both type of metamerism, that is external as well as internal. Whereas in arthropods, metamerism is chiefly external. Externally, the body may be seen divided, but internally they may not show the good metamerism. Okay, so if they are not divided internally, it means the organs, all the organs, or the most of the organs may not be divided internally as well. While main and other vertebrates show an internal metabolism. As I have just given you the example that if you just touch the vertebral column, which is present below the skin, from the morphology, you may not be able to get that our body is segmented. But if you look internally, then you may see that the body is divided. Our muscles are also divided, our nerves are divided, so on and so forth. Even blood vessels are divided, vertebrae and ribs are divided. So it means we have the internal segmentation. So you have seen both the types. The annelids were having both the external metamerism and internal. Arthropod are having the external only, whereas vertebrates are having internal metamerism. Then there is another concept, complete and incomplete metamerism. In annelids, worms, metamerism is complete. Why it is called complete? Because it affects most of the systems. The metamerism are essentially alike. Every segment, if you look at, they look alike. Why they are alike? Because the organs, most of the organs are repeated in it. So called the homonymous type of segments. Okay, so every segment is having blood vessel, every is having ganglia and the nerves, every is having the nephridia, and every is having the coelom and the coelomoderms. So this type of segmentations is complete because it is external as well as internal, and every segment is exactly like the other segment. On the other hand, higher animals such as arthropods and vertebrates shows incomplete metamerism. Because of division of labor, the segments of different regions of their body becomes greatly dissimilar. Now, here you cannot see. If I am giving you one of the example that our vertebral column is showing the segmentation, our ribs are showing the segmentation, but internally, if you look at the organs, they are like the vital organs, the digestive system, the circulatory system, etc. They are not so divided. So this is the so each segment, if you count by counting the number of vertebrae, then you may not find that the rest of the organs are also correspondingly divided in it. So it is the different types of segment. Every segment is different type of segment, so called as heteronomous type of segment. However, incomplete metamerism should not be confused with the repetition of single organ, such as shell plates or gills in certain unsegmented animals, such as mollusks. So, shells on the mollusks, which may show some segmentation, but that is not a incomplete type of segmentation. And accordingly, some lower animals do have the gills repeated, but they are not the incomplete metamerism. Then there is a term pseudometamerism. You must remember and recall the phylum Platyhelminths, where we have discussed the pseudometamerism. Uh, the metamerism shown by annelid is not shown by the platyhelminth, which is called as pseudometamerism, like you have seen in the tapeworm, where strobila were present, the anterior strobila and the middle strobila and the posterior strobila. It refers to superficial segmentation. So the strobila were the superficial segment and could not be termed annulations or the rings or the segments of the annelids. Why they were so called? Because two segments of annelids are laid down in the embryonic stage, whereas proglottids of tapeworm are produced by stabilization. We have discussed that in a tapeworm, the number of segments is from 800 to 900. Why? Because they are continuous. There is a continuous process of formation of new segment at the stabilization. So number is not fixed. Here in the annelids, they are once formed in the embryonic stage, the number which shall remain same up to the adult form, whereas in the tapeworm you will not see. In the young form you will have the less number of stabilization, and then in the adult form you will have more number of stabilizations. So stobila are formed in the tapeworm by budding with the buds remain attached. So it is a separate type of bud, you can say, that is just attached with the anterior one. So if you compare the true metamerism with pseudometamerism, this is the difference that in the true metamerism the number of segments remain fixed, whereas in pseudometamerism they don't have fixed number of segments. Then, in the truly segmented animals, the growth takes place by just elongation of the segments. No more segments are added, whereas in pseudometamerism, growth takes place with the addition of new segments. All segments in the truly segmented animals are of same age because they are formed at the same time and at the same stage of development. Whereas in the proglottids of the platyhelminths, which is showing pseudometamerism, is having a all the segments are differ in the age as well as degree of development because the newly formed will be immature, whereas the earlier formed will be the mature. So they have the different degree of development as well as the age. Segments are functionally interdependent in the two metamerically segmented, though the animal body is segmented, but they work in coordination with one another. So they are dependent upon them. Say, for example, for the locomotion, the muscular contraction repeats in a sequential manner in the annelids, whereas here in the pseudometamerism, each segment is a separate unit. It is independent. It is not dependent upon the anterior one. It may detach from the body. It is just a bud which remain attached with the body. A tapeworm represents a sort of colony of stobila made of linear rows of incomplete individuals. So incomplete individuals are represented by each segment, which are just attached with one another. So you can see it's basically a colony type of feature. And as far as the significance of metamerism is concerned, uh, segments break up the body. So this facilitates the movement, swimming movement. For swimming movement, flexibility of body is required, which is given by the breaking of the body in the form of the segments. Metameres helps in locomotion in several ways. For example, the coordination of muscular action and fluid-filled coelomic compartments. Coelomic compartment you can see because the whole cavity is divided into compartment with the help of the septa. Uh, this coelomic fluid in the compartment as well as the muscles, which are again divided segmentally, they brings efficient swimming and creeping, which is an advancement of ciliary and creeping movement of lower vertebrates. Why efficient? Because a pressure can be put localized in a localized manner because the cavity is divided, muscle is divided. So where the animals wants to put the pressure with the help of the fluid and contract the muscle, only that part will be affected. Rest will not be affected. So in this manner, you have the precise mechanism. Fluid-filled coelomic compartment also provide hydrostatic skeleton for burrowing. So when these animals form the burrow inside the soil, so it is the hydroskeleton. The fluid-filled coelom becomes the hydroskeleton of the animals with the help of which they bore into the soil. Precise movement can takes place by different Turger effect. So as I have already told you, because the segments have divided the body, fluid is divided, muscles are divided, so you can put a precise pressure in a particular organ only, because of the division rest body will not be affected. It will brings the more precise type of locomotion. This is the significance of the. Then another advantage of segmentation is the opportunity for different segments to specialize for different functions. So it is basically meant for division of labor. Once the segments are divided, so one may specialize for one function, other may specialize for other function, thus leading to a rapid evolution of high grade of organization. So segmentation has brought the animals to a high grade of organization. So you can say an advancement in the evolution, with the help of which the animals have become efficient enough. It is not clearly marked in annelids, but is well developed in arthropods. So such type of body organization uh, is more marked in arthropods. That's why the arthropods are more advanced as compared to the annelids. Metamerism has therefore contributed towards the greater complexity of animals. Why complexity of animals? Because the parts are divided. Each part is uh, in turn specializing into a different function. So it is bringing to the complexity of the bodies of the organism, and this complexity is paving the way for rapid evolution of higher organisms, thus some indication of primitiveness of animals. So simply look at the segmentation. If the segmentation is very poor, it means the animal is not highly evolved. It is primitive animal. If the segmentation is good, it is highly evolved. Thanks. This is all about the metamerism.